Good day, students. You are welcome to another video lectures on chemistry of lipids. Under our chemistry of lipid lecture series, today we are going to be discussing steroids and amphiphatic lipids. So steroids are compounds containing a cyclic steroid nucleus or cyclic steroid ring that is called cyclopentanoperhydropenetrine, which is abbreviated as CPPP, C-triple-P. It consists of a phenytoin nucleus that is ring A, B, and C, to which a cyclopentane ring D is attached. So structurally, this is how steroid nucleus look like. So compounds that contain this steroid nucleus are termed steroid compounds or steroids. So steroid nucleus represents a saturated carbons unless when it is specifically shown that it has a double bond. The methyl side chains 19 and 18 attached to carbon 10 and 13, they are shown as single bonds. But at carbon 17, steroid usually contain a side chain. So we said that steroid nucleus is having saturated carbons, and for example, at carbon 17, they usually contain a side chain. We are going to discuss about cholesterol that is having an aliphatic side chain, 8-carbon aliphatic side chain at carbon 17. So steroid in biological system includes, we have some steroids that are occurring in the biological system, which include cholesterol, bile acid, vitamin D, sex hormones, adrenocortical hormones, cytosterols, cardiac glycosides, and alkaloids. These are all examples of steroids that are found in biological system. So when a steroid contains one or more hydroxyl group, it is commonly known as sterol, meaning solid alcohol. So for example, we have cholesterol, and cholesterol is exclusively found in animals. It is the most abundant sterol that is it is found in animals, and it is widely distributed in all cells and is also a major component of the cell membrane and the lipoproteins. Cholesterol comes from a Greek word from chol bile. It means it was first isolated from bile, and cholesterol literally means solid alcohol from bile. So this is the structure of cholesterol, and structurally cholesterol is C27H46O. It has one hydroxyl group at carbon 3. It has one hydroxyl group and a double bond between carbon 5 and 6. And it has 8-carbon aliphatic side chain attached to carbon 17. And cholesterol contains a total of 5 methyl groups. 1, 2, carbon 26 and 27, 1, 2, And we have another one here at carbon 21, 3, carbon 18, 4, and carbon 19 here. All are methyl groups. That makes up 5 methyl groups attached to found in cholesterol. So due to the presence of the OH group at carbon 3, cholesterol is weakly amphiphilic, meaning that when we say a compound or a substance is amphiphilic, it means it has a portion that is hydrophilic and another portion that is lipophilic. So as a structural component of plasma membrane, cholesterol is important. That is important in determining membrane permeability properties because it is found in the cell membrane. It can be used to assess the permeability of that particular membrane. And also the occurrence of cholesterol is much higher in the membrane of subcellular organelles. Cholesterol is also found in association with fatty acids to form what we call cholesterol esters. So cholesterol exists in two forms in the biological system, as cholesterol esters and as free cholesterol. The difference between the two is that in cholesterol esters, the OH is already attached to fatty acid to form an ester in a reaction called esterification reaction to form what we call cholesterol esters. While the free cholesterol, the OH, is intact, ready uh, for esterification, if that is what it is intended for. So the esterification occurs at the OH in carbon 3. So let's look at the properties of cholesterol. And cholesterol, we have the physical properties and we'll also look at the chemical properties. For the physical properties, cholesterol is a yellowish crystalline solid. It's a yellowish crystalline solid that the crystals can be visualized under a microscope, and it shows a notched appearance under the microscope, something like this. This is notched appearance. That's how it shows. The crystals look like under microscope, and it is insoluble in water, but soluble in organic solvents such as the chloroform, benzene, ethyl ether, petroleum ether, ethanol, hexane, and so on and so forth. So these are all organic solvents that cholesterol can be dissolved in them. So for the chemical reaction or chemical properties, cholesterol can undergo several reactions that are useful in qualitative and also quantitative estimation of the cholesterol. So, for example, we have reactions that are called Saalkowski's reaction or Saalkowski's test, Lieberman-Bouchard reaction, and also Zag reaction. And in all of these reactions, they are qualitative tests. It means these are reactions that are used to detect the presence of cholesterol, but not necessarily quantifying the cholesterol. So in Saalkowski's test, it uses chloroform and H2SO4, sulfuric acid, as the reagent that I use in the test. So in terms of Saalkowski's test, So in Sulcocki's test, we have the upper layer containing the chloroform and the lower layer containing the H2SO4. So we have range of colors from bluish red to violet in the upper layer, and also you can find yellow to green color in the H2SO4 layer with green glow that is visible. So in Lieberman-Borchardt reaction, green color or blue color signifies the presence of cholesterol. So what is the function that cholesterol carry out in the body? We have to know that it is a poor conductor of heat and electricity because it has high dielectric constant. It is also present in abundance in nervous tissues. It appears that cholesterol functions as an insulating cover for the transmission of electrical impulses in the nervous system. Cholesterol also performs several other biochemical functions, which include its role in membrane structure and function, and in the synthesis of bile acid, and in the synthesis of hormones and sex hormones and cortical hormones, and also in the synthesis of vitamin D. So the next compound under steroid is what we call aldosterol. Aldosterol occurs in plants. It can also be found as a structural constituent of the membranes that are found in yeast and fungi. And aldosterol is an important precursor for the synthesis of vitamin D. So when aldosterol is exposed to light, the ring of aldosterol, ring B, if you look at it, this is structure of aldosterol. So this is ring A and ring B. So when it is exposed to light, the ring B of the aldosterol opens up and it is converted to argocalciferol, which is a compound containing vitamin D activity. So the other sterols that are present in plant cells include the stigmasterol and the beta-sitosterol, all of which have different biological functions. So we'll quickly look at amphiphatic lipids. As per definition, we can say lipids are insoluble in water, they are hydrophobic. So why are lipids hydrophobic or insoluble in water? This is primarily due to the predominant presence of hydrocarbons group that are found in the lipids. However, some of the lipids possesses polar or hydrophilic groups, which tends to be soluble in water. So molecules which contain both hydrophobic and hydrophilic groups are now termed as amphiphatic molecules. So the general structure of an amphiphatic lipid may be represented as is something or a molecule that has a polar or hydrophilic head and non-polar or hydrophobic tail. So generally, representatively, this is how amphiphatic lipids look like. This is the non-polar tail, okay, and this is the polar head. So examples of amphiphatic lipids among the lipids include the fatty acids, the phospholipids that we already discussed, sphingolipids. We have the bile salt and cholesterol to some extent. They are all amphiphatic in nature. So fatty acid contains hydrocarbon chain with a carboxyl group as functional group. and at physiological pH, the carboxyl group is deprotonated. So the carboxyl group is polar in nature with affinity to water, which is the hydrophilic head now that we are talking about, while the hydrocarbon side chain of the fatty acid is hydrophobic. So if you look at this structure and this carboxylic acid functional group or carboxylate functional group is the hydrophilic head, while the hydrophobic portion is the hydrocarbon side chain, which is the tail. So for phospholipids, they have a hydrophilic head, which is the phosphate group that is attached to choline, ethanolamine, inositol, as the case may be. We have discussed them in our previous lectures. You can always refer back to the previous lectures in phospholipids and glycerophospholipids. And it also has a long hydrophobic tail. So structurally, this is the hydrophobic tail from the glycerol backbone, okay, to the fatty acid hydrocarbon, and the hydrophilic head can be said to be the choline substituent group or the ethanolamine, as the case may be. So let's look at the orientation of these amphiphatic lipids in the membrane. So when amphiphatic lipids are mixed in water, that is the aqueous phase. We have, look at this, this is our aqueous phase, and this is the non-aqueous phase. So the polar groups or the polar heads orient themselves towards the aqueous phase. When they are mixed in water, they tend to aggregate and orient themselves so that the polar head groups aggregate themselves to face outwards, okay, they will face the aqueous medium, while the non-polar tails orient themselves towards the opposite direction, towards the non-aqueous portion. So this leads to the formation of what we call micelles, and the formation of micelle is facilitated by bile salt, which is very important for lipid digestion and absorption. So for lipids to be digested, because they are insoluble in the aqueous medium, they first need to be emulsified by the bile so as to facilitate the formation of these micelle, so that polar portion aggregate themselves towards the aqueous phase and the non-polar aggregate themselves towards the non-aqueous phase. So typically, this is how a micelles looked like. So for the in the case of biological membrane, the lipids occur as a bilayer, or it occurs as a bilayer of lipids forming an oriented molecules having their polar heads towards the outer aqueous phase and the non-polar tails inward to the interior, okay, and both of which are facing either direction. So because it is double layer, it has two layers. You have the extracellular portion and the intracellular portion. So all of which the aqueous phase will be faced by the polar portion, while the non-aqueous phase will be facing inward, and the non-polar portion of the molecules of the lipids are aggregating themselves inward. So in all cases, aqueous phase is having polar head, and look at it, aqueous phase, polar head, and the non-polar head aggregate themselves inward in all the directions. So another component is what we call the liposomes, and liposomes are produced when amphiphatic lipids in aqueous medium are subjected to sonication. You know, the amphiphatic lipids now in aqueous medium, they form micelle, right? So when they are subjected to sonication, and they have what we call intermittent aqueous phases in the liquid bilayer. So when you look at it structurally, liposomes in combination with tissue-specific antigens, they are used as carriers of drugs to target tissue. So structurally, look at it, and this is the bilayer, okay? And you have another orientation inside. You have the aqueous phase inside here, and this is the lipid bilayer, and you have your aqueous phase faced by the polar head, and here intermittently you have the non-polar portion, okay, aggregating themselves, and also here inside you have the core that is aqueous phase, and you have the polar groups also aggregating towards that. So this structure is called a liposome, and it is important in drug delivery system that is used to deliver specific antigens or drug rather to specific tissues. So the next thing we are going to be discussing is emulsion, and emulsions, these are produced when non-polar lipids, for example, triacyl glycerols, are mixed with water. When triacyl glycerols are mixed with water, and they form what we call emulsion, and the particles are larger in size and stabilized by emulsifying agents, usually amphiphatic lipids, such as bile, salt, and phospholipids. So that is all about today's lecture. We are going to stop here. If you are yet to subscribe to the channel, kindly subscribe and put on notification bell so that our next content will be sent, or we will be notified when we upload another content. Thank you for listening and for watching. Kindly subscribe. Thank you. See you next. Bye-bye.